Our project's a lab procedure meant for students in an immunology course at Cal starting next semester. First, we begin with the cell sample preparation to analyze cells from a mouse's spleen and thymus on a microscopic level. We first dissipate the organs into solution, then place them in a conical tube. From there, we centrifuge the tube to mix the solution. When it comes out, there's a pellet at the bottom. Clicking the waste flask, we empty the tube solution and leave only the pellet in the tube. Then we click the lysis buffer, resuspending the pellet in a cloudy solution. The first of 10 questions now appears to test students' comprehension of the lab. There's a scoring system that tells users their score at the end. Input a letter A through D. Now incubate the solution for 2 to 3 minutes. The tube incubates for 2 to 3 minutes. At this point, pipette the suspension to clean the tube and place some cloudy solution in the well plate. Stain the solution sample with blue stain buffer. Now click the conical tube to go to the centrifuge. While the centrifuge does its magic, click the cell count frame and examine the solution in the hemocytometer. Input a letter A through D. Click the pipette man and enter 2.65 milliliters as the correct amount of solution to be added to the conical tube. At this point, click on the antibodies bottle, adding another 15 ml to the solution. And now click Proceed to Staining to move on to the second part of the procedure. To begin this stage, click any of the 4 bottles to add that color solution to the test tubes. Do this 3 times, but make sure not to click the same color twice. Click the Mix button to blend the colors into a cloudy solution. Then click the conical tube to add the original solution into these 3 test tubes. And now the vortex machine will make sure these solutions are well mixed. These tubes incubate in the ice bucket above for 15 to 30 minutes. The test tube incubates for 15 to 30 minutes. Click the PBS bottle to add PBS to the tubes. Then centrifuge the solutions to allow pellets to form at the bottom. Now click the waste flask to leave only the pellets. Add 1% paraformaldehyde to create a small cloudy solution in the tubes. And now we are ready to proceed to flow cytometer. When this final stage appears, click any of the 3 test tubes to find the appropriate data plot. This data differs depending on which 3 colors you chose out of the 4 colored bottles, representing 3 different T-cell proteins. When the Take a Closer Look button appears, click it to analyze the concentrations of various proteins in specific T-cells. Now, the plot allows you to click the top, middle, or bottom portion. Then choose 2 characteristics to analyze for the cells in that portion. Since the top and bottom portions do not contain any T-cells, there's no data for them on the graphs in the next step. If you click the middle portion, however, a graph appears plotting 2 different proteins against each other. The data points each represent a cell, their coordinates determined by the concentration of proteins on those T-cells. In this case, the proteins analyzed here are CD4 and CD8. You can move back a step if you want to take another look. After the last 5 questions of the quiz, 2 extra credit questions appear that add a point each to your score if answered correctly. These are true or false. and after finishing the questions, the lab mascot will tell you your score and recommend you restart the procedure if getting a score lower than 67%.